For this video, I'm going to cover A2 topic 19, genetic technology, and I'm going to focus on learning outcome 19.1C, which requires students to describe the principle of PCR, also known as polymerase chain reaction. And we're going to look into detail about the role of enzyme tag polymerase and other ingredients such as the role of primer and the role of the DNA template. Essay on the principles of PCR actually came out in May, June 17, and you realize that the first two marking point is actually about the introduction about what is the use of PCR. So number one, we can say that PCR is a process to produce large quantity of DNA. Number two, the process is very rapid. So PCR is a rapid process to amplify DNA, and only small sample size is needed. So how many steps does this PCR process requires? As you can see, the process mainly involves part 1, denaturation, part 2, annealing, and part 3, we have replication. So I'm going to show you the sequence in the diagram where number 1, when your DNA is in a double helical form, in a natural in vivo process, which is inside our cell, we will have enzyme helicase to unwind the DNA and break the hydrogen bonds. But in this PCR process, where it's carried out in a thermal cycle, which is a machine, the machine will actually be heated up to 95 degrees Celsius. And this is high temperature enough to denature the DNA, where hydrogen bonds will be broken. Therefore, DNA strands will be separated and the bases will be exposed. So then the next step is the temperature is going to be reduced to 60 to 65 degrees Celsius. And this process is known as annealing, where it only happens at this particular temperature. So who is going to anneal? A primer. A primer is actually a single-stranded DNA that actually has a complementary basis to the desired part of the DNA. So for example, if this is the part of DNA that you want to copy, so you're going to create a short fragment of DNA that when you mix it with your DNA template strand, it will actually bind by complementary base pairing. And the last step will be elongation or replication, where you will see that the DNA will actually get replicated, and this involves enzyme tag polymerase at the temperature of 70 to 75 degrees. And this replication process uses a very special enzyme known as tag polymerase. And this enzyme, as you can see in the essay point, they mention that this tag polymerase is very special as it is heat stable. Therefore, it does not denature easily. And it has high optimum temperature. Therefore, the process will be very efficient as it's constantly running at a very high temperature. So I'll show you in a diagram how does the replication process occurs. So one more importance about the primer here is the enzyme tag polymerase can only carry out the elongation process where there is double-stranded DNA. So the enzyme can only bind to the fragment part where there's 2 strands of DNA, then only it will continue to form phosphodiester bond between the adjacent nucleotide. And this process will continue for 30 to 40 rounds in order to amplify the DNA. So now I'm going to show you the process of joining the assay points. For number 4, we're going to use point 4 and elaborate. So DNA is denatured at 95 degrees Celsius and 2 DNA strands are separated. Then we're going to talk about the annealing, where you're going to join 5, 6 and 7 together. When primer is added, annealing process takes place at 60 to 65 degrees Celsius by complementary base pairing. And the third step involves Taq polymerase, where DNA is replicated at 70 to 75 degrees Celsius. And the process will be repeated for around 30 to 40 times, therefore your DNA concentration will increase. And take note, when you talk about the role of DNA polymerase, there are 2 main features which we're going to see how you apply this in answering structure questions. This question on PCR came out in October-November 16, paper 41. So you will see that the question actually gave you the step, but they wanted you to be able to analyze what is the importance of each step. Therefore, the question started with why the DNA strand is heated to 95. So if you know only denaturation, so what else can you elaborate? So we know that it's to separate the 2 DNA strands by denaturation. So secondly, what you can elaborate here is, as the DNA denature, what is actually happening? Hydrogen bonds between the bases are broken. Then to continue, in case question change to 3 marker, you will say that the bases are exposed, therefore producing a template strand for complementary base pairing. Question number 2, why primers are added? So as you know that the primers will anneal in the second step, so here we will actually get marked if you mention what is actually a primer and what do they do. So we can say that primer anneals to the DNA template strand by complementary base pairing. So this is actually not really the role, but they actually award you for knowing that primer is a short fragment of DNA that is single-stranded, therefore able to bind to the template strand by complementary base pairing. And this is very specific. So we will say that at a specific section of DNA that you would want to amplify. So then now we are focusing on the 2 main roles of the primer. Why is it necessary? So another point here, we will say that because DNA polymerase only attaches to the double-stranded DNA. And another main role here is, we know that when your primer is actually added, so it prevents the DNA from forming hydrogen bonds again. So therefore, in other words, it's like preventing the DNA from re-annealing. If not, your process of denaturation will be wasted, because as they re-anneal, the double-stranded will rewind and you wouldn't have the template to be replicated. Okay, so the last question here involves the role of tag polymerase. So we will explain what is the role of polymerase and why do we need to use this particular type of enzyme. So the role of polymerase is to synthesize a complementary DNA strand, and then the next thing will be, why must be tag polymerase? So the tag actually comes from a bacterial name where the bacteria actually survives in the hot spring. Therefore, this enzyme has a high optimum temperature and heat stable, or you can call it thermal stable. So the enzyme does not denature easily. Therefore, does not require to be replaced after every cycle. And finally, as you know that the enzyme has a higher optimum temperature and the cycle is constantly running at a high temperature, therefore this process of replication will be more efficient. So take note, this more efficient is because of more ES complex, more kinetic energy, therefore higher rate of enzyme reaction. If you would like to know more about the application of PCR in genetic fingerprinting, in electrophoresis, or in a microarray, do not hesitate to contact me for group or private tuition for more interesting lesson. Thank you.
That's all for today's video. If you are interested in more genuine sharing by other geniuses, please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with one or two past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. GenieUs Hub will get genius teachers to fulfill your request for the solution. Genie will see you next time. Bye-bye.